self-explanatory, but we're going to talk about kind of the embryology of the development of the skeletal system. And hopefully, I hope you're learning things when you're studying the bones and you realize how individual and unique our skeletons are. Even though we have the same plan and we have the same bones, there is so much about our skeleton that gives us uniquely who we are. And the nice thing about studying the skeleton first in a class like this is that all the other organs and systems that we learn are going to be based upon the skeleton. So once you have learned these words and the way to describe the skeleton, everything else will begin to fall in place. And I can't say that enough. I really want you to understand that. Because anatomy, you know, the biggest thing I hear everybody, I'm so afraid of memorizing. I'm so afraid of I won't be able to keep up. And you know, I really say throw memorization out the door. Don't even try to go there. What you need to learn is the language. And yes, there is a certain amount of memorization in learning any language. But once you have that language, then everything else will become so much easier. And then, of course, aging of the skeletal system. We'll talk about that too. So there are about 270 bones at birth and about 206 in the adult skeleton. So what happened to all those other 64 bones?
Fused.
Right, so they were fused. So like, for instance, the occipital bone at birth is 5 different bones, but in the adult it's 1 bone. Same for the sternum, same for the sacrum, so I hope you will kind of understand that. And then the way that these, let me see, what do I want to say? These ossicles, the bones fuse. Our bones, up until puberty, there's really not very much difference between males and females. In fact, in a court of law, we would never attempt to determine sex of an individual based upon their skeleton if it was before the age of puberty. Because after puberty, next week when you study the inferior appendicular skeleton, you'll see, you know, and be able to really, it will really come home to you how different the skeletons are based on the pelvis. Now, there are skull differences, but they're not enough that we could actually accurately sex anyone by their skull. We can say likely if you had no pelvic and only have the skull, then you can do a likely, you can give a sex likely to that bone, but it's not totally. And of course, the bone growth is not complete until age 20 to 25, and we have, if you didn't see it already in the lab, the skeleton that is over by the elephant skull, you can find it's in the cabinet, that is a really young skeleton. So when I'm talking today about the epiphyseal plates and about growth plates, make sure that you look at that skeleton, because that's probably our youngest skeleton that is easy for you to see, besides the juvenile skeleton that we have of the little 6-year-old. But that skeleton, you can see these epiphyseal plates. So most bones are totally fused, totally grown. You are your adult skeleton by the age of 25. Usually 23 to 25 is when the skeleton totally fuses and you are completely unique. And of course, then you start the aging process, which you probably don't want to hear when you're 25, but it's true. And but our skeleton is constantly being remodeled. I mean, think about when you break a bone, it's healed in like 6 weeks. So bone is rapidly growing tissue, and it's constantly being remodeled. People that are on bed rest for a certain amount of time, I mean, their bones will actually thin. I mean, and this is something they've discovered from astronauts too, because they don't have that weight bearing. Up in space, there's no weight, so the skeleton actually begins to lose tissue. You lose density of bone right away if you're not using it. And then, you know, remodeling of the bone. We see these different markings on the bone. You've probably seen enough already to notice that some bones are rougher than others, some articulation sites are easier to see than others. And this is all remodeling that is happening based upon that individual's life. And then of course, and this happens all through your life. So say you've never ridden a motorcycle before and suddenly you start riding a motorcycle to and from work for the next 10 years, you're probably going to have some spurs here on your ischial tuberosity. And if someone found your skeleton, they're going to say, well, it looks like this person might have had some kind of jarring action sitting down, most likely maybe motorcycle riding. So we can tell these things. I mean, you see it on CSI all the time, right? You see and this is how they do this. It's all the studies and research that have been done on this remodeling process. It's very fascinating. And then, of course, why would a person become shorter? Well, I think it says that. Because of this compression. So our vertebrae begin to compress, the discs begin to compress. So our skeleton is constantly changing forever. And then this is just kind of a little schematic of the development of the skeletal system. And you can see here what we've kind of seen last week, this 4-week-old embryo. And you can see the somites and the beginning of the vertebral bodies there. And then you can see the limb buds beginning to poke out. And then finally, an 8-week-old embryo or fetus is beginning to develop those limbs. And so this is just this gradual process. Now, I'm not going into the cell molecular parts of embryology because I just get in trouble. I get myself in trouble. But you can see here sort of this process of the limbs forming. Now, something that's important here, the important part of this, is that this mesenchyme, the buds invaded by mesenchyme, which comes from the mesoderm, and the mesenchyme gives rise to the cartilage. So our bones are formed in cartilage first, and then that cartilage is then calcified and turned into actual bone. And I have some images where I can show you the cartilage plates as they're forming. So this is kind of interesting. So all of these shapes would then first be cartilage and the bone. And when you saw the fetal skeleton already, you saw that a lot of the bone is still cartilage. And so this is an important thing to notice. And we'll talk, there's 2 different types of bone growth that we have in humans. And we'll talk about those different kinds. And then this is just this origin of the axial skeleton, the notochord, which gives rise then to the vertebral bodies. And the somites, and again, this is this process of the notochord being replaced by the vertebrae. And the nucleus pulposa, do you guys remember what that is? What is the nucleus pulposa?
Inside the discs.
It was from lab 1.
It's the inside of the disc.
Right, the nucleus pulposa is that kind of gelatinous disc that's inside the fibrous cartilage disc that is in between each vertebrae. So that nucleus pulposa is sort of this remnant of the notochord in development, which is kind of interesting. And we call these joints between the vertebrae, we call them symphysis. We have a symphysis that you should have looked at this week or will look at, which is right here where the clavicles join to the sternum. This is a symphysis joint. And then we have another symphysis joint that we'll look at next week in lab, the pubic symphysis between the 2 pubis bones. And something that's just kind of an interesting pattern, all of these symphysis joints are located down the midline axis of the body. Which is just kind of an unusual pattern to be human. And again, this is another picture of this neurulation. So you're seeing a cross section between the developing embryo, and you can see here then this neural tube, and you can kind of begin to see the mesoderm filling in. You can kind of begin to see this development of the structure of the bones and of our brain and of our spinal cord. And then just to give you a little comparison of the adult to fetal skull. So you can see that, fetal skull is in many parts. You guys have already had this from lab last week. But if anyone has any questions, now's your time to ask about this. And these are the fontanelles. So why do we have fontanelles in the skull? What's the purpose of the fontanelles? Right, so the baby can pass through the birth canal.
Also when the skull grows really fast, then it can, yeah.
Right, more importantly though than even the birth canal is what you just said, this rapid brain growth. I mean, from birth to a year, our brain doubles in size. And if you didn't see that in the lab, ask someone to show you the fetal skull and the 1-year-old skull, and you can see just tremendous growth. And if we didn't have these fontanelles, in fact, it is a pathology where sometimes these fontanelles close up too soon, and it causes pretty severe mental, retardation and problems. So this can be a real serious issue if these are not open. But having this open allows for this rapid brain growth. And the skull, the bones of the skull develop differently than the rest of the body. So after I told you that we form our bones in cartilage and then that is ossified, and I'll show you pictures of how that happens. This bones of our skull develop differently. They're called, it's a membranous ossification. And so what happens in the fetal development is that the bones kind of ossify and develop out in a flat membranous sort of sheath. So this fontanelle ossifies. So it's a little bit, it's slightly different type of growth, which will make more sense as when I show you more pictures of it. So it's kind of interesting, I think, that, and that going back to this slide, the neurulation, I think I told you before how facial muscles and everything is a little bit different. So cranium up, a little bit different process from the postcranial skeleton. And here's just another picture of this fetal skull. I'm going to buzz by some of this kind of quick. And then this is just a comparison of animals and to skulls. Anyone have any questions about animal skulls and humans? Okay, so here is this talking about the skeleton continually develops and remodels throughout life. And this Haverson system, which I'm going to show you pictures of how our bones are developed in this Haverson system. But we classify the bones by 3 different, we say that we have an axial skeleton, which you're familiar with. Then we have the appendicular skeleton, which you've had this week and next week. And then we have the brachial skeleton. And the brachial skeleton is formed from the gill arches. We have these vestigial gill arches. And that's kind of what you're seeing a picture of here. The inner ear bones development, some of these, those are just ossification. But when you see the bones develop fetally, it's just a sort of a different type of development. Let me show you this next slide. Okay, so here, and I have another picture of the brachial skeleton. So this would be the axial and appendicular skeleton. These are things you've already looked at in lab. This week we're working on the pectoral girdle. Next week we'll work on the pelvic girdle. Does everyone understand what those are? The girdle is a circle of bone that circles around the axial skeleton. And then this is the brachial skeleton. Oh, I had another picture, but it was in the keynote slide, sorry. So the brachial skeleton, so evolutionarily, we evolved, vertebrates all evolved from this common ancestor, and or from a common ancestor. And when you look at the embryological development of animals, you can see vertebrates go through similar types of structures. And the brachial skeleton are these gill arches. And we have this Meckel's cartilage, the styloid process, which is part of the temporal bone. The styloid hyoid ligament, so that's a ligament, no longer a bone. The lesser horn of the hyoid bone, the greater horn, the thyroid cartilage, cricoid cartilage, and tracheal rings. So these, And I have this great picture of a fish you could see, but it's in my other show. But so if you saw a fish and their gills, it's located approximately in the same place, and these develop from those structures embryonically that in a fish would form gill arches. As the embryo develop, if it's a human, these develop into these bones and these structures. Does everyone understand what I mean by that? Okay, and so then just comparison-wise, we'll look here at a cat, and very similar to humans. I mean, you can see here, those of you that already had lab and those that haven't. So here is the humerus. We have the humerus. Here is the ulna. Here is the olecranon process, the little point on your elbow. The cat has one too. Here would be the radius, right here. And you can even see, if you look at the cat, you'll even see that the structure of this appendicular joint is the same with the capitulum and the trochlea, but they don't have the ability to pronate and supinate. But if you look at any of, like the pig skeleton or any of these other skeletons, you'll see these same bones with the same processes, but just a little bit different type of articulation. And of course there are, like the clavicle on the cat is mostly cartilage, very little bone, so it's very flexible. Here's a good one to show you, kind of some homologous structures, but that are very different. Okay, so here in a bird, so those of you that have had lab already, and those of you who haven't, pay attention. Where is the coracoid process? Who's had lab already can say it. Coracoid process. Coracoid. No, it's from this week. It's from this appendicular skeleton. Scapula. It's on the scapula. Right. So the coracoid process, I have one here, is a little bone that is protrudes from, we have the acromion process, and it's this little process that protrudes under the scapula. And we have the biceps attaches there, we have 3 different muscles that attach there. It's a very, very important structure for us, muscularly, but it's connected to the scapula. But now on a chicken, or a pigeon, as the case may be, there is the little bone, I think it's right here, the little coracoid bone. So they have an actual bone. Let me see if I can pinpoint it. I've got to get a laser pointer. It's right here. Can you guys see that little bone? And it kind of, the scapula of a chicken and a bird is a little thin bone right here. And then here's the coracoid bone in a bird. And then, of course, this, what bone is this? Homologous. The sternum. So here's the sternum. Now the wishbone, we're all familiar with the wishbone of a chicken, that's the clavicle. Yeah, we're homologous to our clavicle. So birds, so to just get a picture of the differences in morphology, so here's this coracoid process on us, but it's a whole bone on a chicken or a bird. And so it's kind of helps to, and if you think about the bird, I mean, this middle section of the bird needs to be, very stiff, like a fuselage of a jet plane. You don't want it to be flexing. And so they have these, the whole structure, their ribs, everything is built to be lightweight and this firm structure and this coracoid bone. So, and coracoid, by the way, means raven's beak. And I always think that's easy to remember because you find it as a bone in a bird. And it looks like a bird's beak when you see it in the human. It's a pretty hefty little process. If you didn't notice it, make sure you do notice it. It's an important structure since 3 muscles attached to it. Okay, so let's talk for a minute about the types of bones. Would have been nice to have gotten this before you had your first lab, but anyway, but we're here having it now. So we have long bones or short bones, flat bones, irregular, and sesamoid bones. So long bones are defined as bones that are longer than they are wide. So arm, the bones of your brachium and antebrachium, your fingers. These are long bones. My distal flange 2, right here, that's a long bone, even though the bone itself is, less than ½ an inch. But it is longer than it is wide. And there's a definite special structure to those type of long bones. Now, a short bone would be like the carpal bones. Those would be short.